you. Thanks. Thank you so much. Hey, everyone. It's really nice to be here. Super excited to be talking to all of you, and thanks for the invitation. So what I've done today is I just have a few slides, actually. It's just pictures, and I'd like to share the story of my life, how I have come about in biotechnology and the things I'm doing with biotech. And I really want to kind of give an idea of the types of things people can do with biology. I was here for the previous talk and just inspired by what you can do with biological systems, and kind of taking it from the medical field towards more of can we make things using biology that are relevant to all of us. And I will talk a little more detail about the Impossible Burger, and would be really curious to hear how many people here have tried it and things like that. But let me just go through and walk you through my sort of journey, my background, and so it'll be woven into the rest of the presentation here. One thing that I struggled with was saying, so what's my role today? Like I mean, I can put a title, but am I a scientist? Am I a technologist? And I was like, I'm actually doing a little bit of everything. I started my career as a scientist. But then I eventually went into becoming more of a technologist. What I mean by that is taking the science and applying it in large scale and using engineering to actually produce something in manufacturing scale. And currently, as Itzel mentioned, I'm more in the business, which means now that I'm making these things, I'm figuring out how to sell them and how to, what it takes to make this economically feasible. And I will end with why this is very important, especially with some of the challenges facing the world today, like climate change and global warming, and where sustainability is extremely important, finding ways to make things not by burning only fossil fuels is extremely important. So I'll get into that a little bit, and hopefully I'll get questions then. Okay, so I just wanted to start with sort of my little bit of my history. So I grew up, I was born and brought up in India. I grew up in the beautiful city of Delhi, very crowded city. That's a very nice picture. It can get really crowded very fast. I went through my elementary, middle, high school in Delhi. The education system in India is very different from how it is in the US. I now have a middle school, a daughter in middle school, and sometimes I struggle to understand how things are done here, but it's really interesting. I think one of the amazing things about the American education system is how open the system is to kids trying out new things and experimenting with new things through school, through college. In India, when you are in 9th grade, you have to commit to choosing certain languages, and when you're in 11th grade, you pretty much have to commit to, are you going to go in the science field or something else? Okay, so 11th is when you have to make that choice. So in 11th grade, if you choose to go after humanities or commerce or accounting or something else, you cannot go back to science later on in life. So it's a very critical decision that's made at a very young age and can really go wrong. So I mean, I'm really glad I actually chose to go into science in 11th grade because I loved it. And so I want to talk about how that kind of love for science has shaped what I did with the rest of my career. So for my undergraduate and master's, I went to the All India Institute of Medical Sciences, which is a premier institute in India, in Delhi. It's a medical campus. They also had courses in human biology and biotechnology. So that's what I studied. I was an undergrad major in human biology and biophysics. What that means is kind of taking the medical perspective, which is like, what's the human body, how does it function, what are all the systems, but focusing more on the details of the biology rather than sort of the healthcare and patient side of things. So truly understanding at sort of the cellular level, at the biochemical level, what is going on inside human bodies. And around the time that I was doing my undergrad and my master's is when the Human Genome Project was a big deal. So the Human Genome Project was initiated in the 90s, and towards the end of the 90s, when I was in my undergrad, is when a lot of new information was coming from that. What that means is people actually took the DNA from humans and were able to really try to start decoding what the different DNA sequences are coding for. It was an extremely exciting time to be thinking about biology. And so that's what got me really interested in thinking about this element that, you can make things using biology that don't just, that don't necessarily exist the way they are in nature. Let me explain what I mean by that. So I don't know if anyone here has heard the term recombinant DNA technology. Have people heard that term? Cool. So what that literally means is we now have the ability to take pieces of DNA from one organism and put that into a different organism and create sort of novel things which don't necessarily exist in nature, okay? Why would you need that? So think of the example of diabetes, right? When people have diabetes, they have a sugar regulation problem in the body. What manages sugar level in the body is the hormone called insulin, okay? And so that's produced by pancreas in all animals, including humans. And for the longest time, when people had diabetes, the deficiencies in insulin, they needed to be provided insulin. This came from animals. So what we did was we were extracting pancreas from animals and extracting the insulin, and that was the medicine that was being supplied to people who had diabetes. With the advent of something like recombinant DNA technology, what people could do was figure out the part of the DNA that codes for this protein, which is insulin. Take out that piece of DNA and plug that into the DNA of another organism, like a bacteria or a yeast, and I'll get into why that's important. These are extremely small organisms which you can manipulate, which you can grow really fast. We can grow in controlled environments, and you don't have to go slaughter animals to get this product. Insulin was one of the breakthroughs of using this recombinant DNA technology, where we were able to take the gene, put it into bacteria, produce it at large scale. And today, that's how insulin is produced for treating diabetes, okay? So when I was in my master's, this is the kind of thing that I was reading about. The Human Genome Project was happening, and I realized that that's what fascinated me the most. I still did not know what exactly I wanted to do, or it wasn't like a very well thought out career path, just that I was really enjoying science and math, and I was really enjoying biology in particular. So following my master's, I came to the US to pursue a PhD in biochemistry, again, because I realized that what really interests me is the biological systems. And so I went to Cornell in Ithaca, which is just beautiful, stunning, very different from Delhi. The first few nights I couldn't sleep because it was too quiet. It was like, where is all the noise? But got used to that, and also got used to having snow for 6 months in a year. So it's all great. Beautiful campus. And at Cornell, what I ended up doing was working with basic biological systems. So I love this cartoon. Let me see if I can kind of point here. So, okay, so what this is showing is just like a very fun cartoon of showing what's happening inside a cell, okay? So here you have DNA, which is the building block of life. Are people here familiar? Has anyone heard the term central dogma of biology? Okay, yeah, quite a few hands there. So the fact that information in biological systems is coded in DNA, that gets read into what's called an RNA, which you think of sort of an intermediate molecule, and then that makes proteins. And a lot of what's happening inside our bodies, inside our systems, in plants and animals, in any living organism, is actually happening due to proteins. They are they maintain the structure of your cells, they maintain functionality, they are the enzymes which carry out critical functions, okay? So this is kind of showing that central dogma in some way that, the DNA is being read, you're forming an RNA, the RNA is then making proteins, you're showing the amino acids here, and this kind of complex structure. So in my PhD, I actually spent 6 years just understanding one of these little pieces from one organism. And that's what it took, 6 years to study all of that. But the important skill that I would say that I learned, whether it was in my master's or in my PhD, was the ability to learn. Because things keep coming, others like new information coming all the time. And especially now with genomics, where everything's getting sequenced at a rapid rate, it's really easy, it's very cheap, a lot of people are doing it. You have AI, which enables you to read information at a speed that no human could and no computer previously could. So now we are generating, so it's not just a string of letters, but that the string of letters is actually now making sense, right? So you're taking data and you're converting it to knowledge at a rate which is really, really fast. And so one of the things for me was kind of like really getting into the system and saying, okay, how does this work? How do cells work? So I did my PhD in a very specific subject, and it was understanding the process of going from DNA to RNA. It's called transcription. So I spent a bunch of time figuring that out and really enjoyed what I learned at the stage. And then from there, I came to California, and I came to University of California, San Francisco. It's the Mission Bay campus, where I pursued my postdoctoral work. And so the way it works is if you are interested in an academic career, which means if you want to become a professor at a university, you get your PhD, you go do postdoctoral work, and that's your first time doing independent research. So in the PhD, it's the professor who's kind of defining sort of the big picture organization of the project, and then you kind of come in with your independent idea. As a postdoc, you find your own source of money, and you go and explore your own project. So I was working here. I had a Leukemia and Lymphoma Society fellowship, and the reason was that I was studying something. I was studying a system in cells, which was identical between the way the system works in yeast and in humans, and it actually plays a role in cancer indication. And so that's why I was funded by Leukemia and Lymphoma Society. What I did in my postdoc work now was I went from understanding things purely at mechanistic level or very biochemistry level to thinking about the organism as a whole, okay? And so I was doing a lot of work with genetics, so yeast genetics, bacterial genetics. And yeast and bacteria are extremely powerful, okay? So they are called model organisms. There are some types of organisms called model organisms. Maybe you guys have heard of them, like E. coli, yeast, the one that's used for baking. It's called Saccharomyces cerevisiae. There is another one called Schizosaccharomyces pombe. These are all organisms that people have studied for decades. So what's powerful about them? First, they are unicellular. So it's one cell. And fundamentally, the organelles in the cell and the sort of overall organization of the information is the same, highly conserved evolutionarily from bacteria through larger organisms all the way to humans. So if you can understand things in these model organisms, a lot of the time you can predict what's happening at larger scale. And so for me, it was very useful now to sort of understand how organisms work as a whole. So I almost, I used to joke that, I went from, I'm going up the evolutionary ladder, like, starting to just understand biochemistry, then work on bacteria, and then work on yeast. So the obvious next step was go towards, plants and humans and animals. So this was a really critical time for me because until this point, was very convinced that I wanted to become a professor and wanted to pursue an academic path. However, in 2011, what happened was I was introduced to this company called Codexis. And I started talking to people who were using information they were learning, knowledge they were learning in schools, colleges, to apply and make products, like make real things that impacted people. And the more I talked to people like this, the more I went and visited these companies, I actually felt very inspired that I really like teaching, I really like the academic path, but I really want to be more in touch with making something. So what they do at Codexis is they basically take proteins or enzymes which naturally exist, and they make them better, right? So they have, let's say an enzyme, what does an enzyme do? It catalyzes reactions going from A to B. So you have a substrate, the enzyme catalyzes it, and you have a product, okay? these enzymes can be made to be more productive. You can have faster, you can have it more efficient, you can reduce byproducts, you can make it more sustainable. So Codexis was a company where we were manipulating enzymes and doing protein engineering to have what's called green chemistry, so more sustainable enzymes. In pharmaceuticals, I'm showing you a picture of pills. A lot of the times you're using enzymes, they come from chemical processes, and your biological processes can be a lot less harmful to the environment. So we were working on that, and so once I heard about Codexis and I went and met them, I really liked it, so I was like, okay, I'm gonna move. And it was a very hard decision for me. It was a very hard decision for my professor. He didn't like it, but we eventually came around that this is the right thing for me to do. And so I spent a few years here understanding how people use microorganisms to produce actual products, okay? So just spent a few years here, and then eventually I went to Impossible. So I'm going to focus mostly on Impossible because I think that's sort of the most exciting part of my story. I've been there for, I was there for almost a decade, and I think I'd love to hear what people here have to say about the Impossible Burger. So first, before I talk more, I just want to know how many people here have tasted the product. Awesome, awesome. Have people tasted the burger, or have people tasted sausage like at the Starbucks? Okay, great, great. Okay, so Impossible happened to me also almost by accident. Someone I knew who was working at Codexis moved to Impossible. And so here's the really funny story about Impossible. The company was founded in 2011 by Pat Brown. Pat was a professor at Stanford, and he'd actually done some really groundbreaking work in biochemistry in the past. You wouldn't think a biochemistry and MD, PhD is going to sort of make food, So when I heard about the company and when they reached out to me, I went and talked to Pat, and I was like, why is a scientist thinking about food? And he made a couple of really interesting observations that when we think about making new foods, or at least this is what I used to think 10 years back, you think about like, I'm gonna add more salt, add less garlic, fry this way, cook this way. But he was kind of taking this really big step back and say, how is food made, right? So if you think about it, we're growing grass, we're putting a ton of water, growing grass, having the cows eat the grass, and then the cow grows for X number of years, and then we slaughter the cow, and we get the meat. For this much meat, the amount of energy and efficiency that goes into the system, it's mind-boggling, okay? And one of the things he said, I remember this so distinctly, he's like, and then of course there is the whole global warming, climate change impact of animal farming. So you have a lot of cows. Back in the day, if it was just some cows grazing, it's different. Factory farming is where you're just growing cows with the purpose of making food. And so much of the world's land and water and all these systems currently are diverted to factory farming, okay? And then the cows, of course, produce a lot of methane. They're burping and farting all the time. And that's literally one of the top causes of greenhouse gas emissions right now, okay? But here's what Pat said. He's like, look, you can't make people feel guilty about global warming and say, don't eat meat. You have to find an alternative. You have to put something that people like and enjoy, right? And they have to enjoy it, and that's how, and you create an alternative product, and they should want to buy it because it tastes good. And I was sold when I heard that. So the approach we took at Impossible was understand what makes meat meat at the molecular level, at the scientific level, and then reverse engineer that and see, can we make that without the animal? If I can understand at a basic level what it is, then I can make it. So it's kind of taking the science principles, which is figuring out what's in it, and then combining it with the engineering principles, which is now how do I make it, okay? So this is what our raw product looks like. And one of the big differences between, this was one of the first products in 2015, 2016 when we launched. If you compared this to before that, any other non-animal product, right? So all the plant-based things, were like garden burgers and, they don't look like meat, and they're not meant to. The audience is vegetarians. It's people like me who grew up eating vegetarian food their whole life, and so when I go to a barbecue, I needed like a sorry excuse of a burger on the side. So that's what it was for me. But this is for people who like meat, right? So they enjoy it. So we had to make it look, taste like meat. And the reason it looks like meat is because of the texture and the color, right? So you see that pink color, it looks like blood. And so that was, this blood was the focus of my work, because otherwise I find it hard to kind of see how my entire background justified sort of being in a food company. And the way it justified is I led the team that figured out how to make this protein. It's called heme, okay? So in all our bodies, in our blood, we have a hemoglobin. I'm sure you are all familiar with that. Hemoglobin is a fascinating protein. It has iron bound to the protein. It has a heme group. It's a small heterocyclic group, iron bound to it, bound to the protein. And it gives it this very bright red color. That's what makes your blood red. And so we figured out that if we can make hemoglobin without the animal, the only way you can currently get hemoglobin anywhere is from animals and human in the blood, So can we make hemoglobin without touching an animal? And the answer is yes, right? So that's the work that I focused on. So what we did was we found a protein that's naturally found in plants, in soybean plants, actually. So if you went into the field and pulled out a soybean plant, the roots of the plant have these little tiny nodules. You cut them open, they're actually bright red in color, which you wouldn't expect because plants don't have blood, but this one does. And so that protein is what we decided we would make to put into the burger. And what we had to do was figure out how to use a model organism like bacteria or yeast to make heme. So we ended up using yeast. It was a very, I want to say, very hard project for a few years, where what we had to do was two things. One, we had to take a foreign protein, we put it into the yeast and have it grow and produce something. But two, we had to manipulate the yeast itself to produce more of the heme factor, right? So there is metabolic changes, there is cellular changes, and we had to do all of them together. So in these years, what I ended up doing, in addition to being a scientist, was actually hiring people, building a team, and recognizing the different types of scientists that exist, like for what kind of work do I need a molecular biologist? Where do I need a biochemist? Where do I need a chemist, an analytical chemist? Where do I need an engineer? Where is it a chemical engineer? Where is it a software engineer? Where is it an electrical or a mechanical engineer? So figuring out all those things was something I really enjoyed doing in the course of my time at Impossible. So the fundamental concept was the company had sort of, it had texture team and a flavor team, and we had these groups of people. We used to do tours in the lab, actually, and we used to have schools come through because it was really fascinating. When you have to reverse engineer, for instance, what does meat taste like? One of the things you want to know is how do your teeth feel when you chew meat? And so we found a way to, we found a small device that we could make in the lab, which could put pressure on the meat and give us a number that kind of predicted how your teeth would chew something, right? Similarly, for flavor, we reverse engineered, we took meat, we took all the components of meat, extracted them, and we put them through what's called chromatography mass spectrometry. It means you separate molecules and it identifies each one of them by mass. And then we would have one person literally sitting there smelling as these things were coming out through the pipes, and he would smell and be like, that smells like socks. We literally had and it was so funny because once we had a media tour coming through the lab and someone was saying, that smells like dirty socks. And the reporters just froze, and we were like, it's just like it smells like, there are all these things which are, in fact, if you did that with real beef, you get these complex mixtures. Each one of them have completely different smells. Anyway, so we used all these systems, reverse engineered overall the product on one hand, and then on the other hand, we made heme separately. And in the course of this, I went from figuring things out in a lab, which was amazing, scientifically challenging, intellectually challenging, but kind of going from there into the real world, which is great, I can make this product, who's going to buy this? And can I make the product for less money than how much I sell it for? And that's really, really hard, because you can hire 50 people and scientists and engineers, give them all the money, and they can do all the work. But can you do that with lesser and lesser? Because someone needs to buy it at your Safeway or Target or Walmart, whatever. So that's when the reality for me hit, and that's what I realized I'm the most excited about. So here I was starting as a scientist, but concluding that actually business is what really, really motivates me. I love using science to move business. And so here I'm showing you a picture of what a small-scale plant looks like. On the left, the tank, you see that woman standing next to the tank, it's probably like a few thousand liter. I am standing here next to a tank that you can see the top of. It's several 100,000 liters. It's about 4 to 5 stories long. And we are producing food in that tank. So we're producing food proteins in that tank. So imagine cleaning that, right? Imagine, and you have to clean it between every run, okay? So every time I take my bacteria, my yeast, which I have modified, I put it in the tank, I grow it for a certain number of hours. I have to provide food to the bugs to grow, to the organisms, so I have to provide salt, water, sugar, minerals, vitamins. And then after a certain amount of time, it uses all this raw material, and I have made the genetic changes, so it produces what I want it to produce. And then I take out the product, extract what I want, which is the heme protein, and I have to clean this thing. So imagine when you're cleaning in your sink or like a small pan, if a bigger pan gets harder, if it's got all these nooks and crannies, it's even harder. We have to clean this and get it ready for the next, and these are all borderline sterile processes. So, and you have to do that without spending too much money. So it's fascinating to figure out how to do something like this. So to me, understanding sort of at the DNA level, the system, and then thinking, how do I get this to be made in a way that I can actually sell has been the sort of the most fascinating part of my journey. So I know I didn't get into a lot of details with sort of the science aspect of it, but I just want to end with something for you guys, okay? So if you were to choose a career, say in biology, biotechnology, what can you do? So I did this very basic, I call it first principles experiment, which is, look at everything around you, right? Where do things come from? Like, how are they made, right? That curiosity is what drove me to think about how is I'm wearing this, like where's this fabric coming from? Where's this wood coming from? Where are different things coming from? And you will see that a lot of the things around us in the world are coming from highly unsustainable processes, because we have built the world around sort of things that come from petrol, fossil fuels. And biology now holds the promise of being able to make a lot of these things in much better ways. So I'm just showing examples of things which are currently made using biotech, and I hope that you guys would consider something like this, right? So of course, medicines, a lot of medicines today come through biotech processes. Medicines are expensive, so it can afford the biotech processes. So no questions there, no one challenges how to use biology. But if you start talking about fertilizers, okay? Currently fertilizers come from petroleum. So the process of making fertilizer is highly unsustainable. Very cheap, so people keep doing it even if it's unsustainable. But the other piece is it's polluting the environment with chemicals. And you add it in the soil, and then the water runs off, that goes into the rivers. There's like chemicals and plastics in the system. And so if we could switch to a biology-based system, you could bypass a lot of these issues. And today, again, with the genomics information and with the kind of AI tools which are out there, we see progress happening at a crazy rate. So by the time some of you are making sort of your graduate school, postgraduate, or career business decisions, we're going to be in a much better place even to use these tools and apply them to solve real problems. So This is something I really enjoy. That's a petri dish meat. I think some of you may have heard that people have tried to culture even animal cells. Very hard to do. And currently not cost-wise, it doesn't make sense, right? I think one burger will be like, I don't know, thousands of dollars. But it's, we've improved. It used to be tens of thousands of dollars. So it's come down. But, when will it hit $4.99 at Walmart? I don't know. But yeah, I just hope that everyone sees sort of the potential in what can be done with biology, and I'm happy to take any questions.
After developing the Impossible Burger, what's the next product you're looking to develop?
So, good question. So I actually left Impossible about a year back, and now what I'm doing is I'm working on the factory side, on the business side, and I'm working with many different people who are trying to make different types of proteins. So some of them are food proteins, for instance. There are people who are trying to make milk, or people who are trying to make egg, also using biotech. There are people who are making medicines, enzymes, and so it's because I'm on sort of the engineering side of things, I'm working with a lot of different products right now. Thank you. Yes.
So I see that when making the meat, trying to replicate the flavor, because that's what the meat that's what they've always had, the exact flavor. What about the myoglobin? Is there a way to make artificial myoglobin to make the flavor correct?
Yes, that's a good question. So just for everyone, the hemoglobin in the human blood, the similar protein that's found in muscles in cow, is called myoglobin. And so we could have chosen to use myoglobin. We chose to use leg hemoglobin, which is a hemoglobin from a plant, and any of them can actually add different flavors. In our product, leg hemoglobin gave the best performance in terms of we liked the taste and the overall functionality the best.